let's start with this 2011 paper. Okay, what are we looking at here?
We are looking at the ciliated epithelium of the trachea.
Good, so what can you see?
I can see the cilia present, B.
Yep.
I can see the goblet cells, I think, there's A.
Okay.
And then mucus glands, maybe.
Okay.
Yeah.
Looking at A then, what do you think this is filled with?
Mucin.
Yeah,
Mucus.
good. So you can see the droplets, they're quite clearly spherical.
Yeah.
And we're looking at an EM. All right, go ahead, calculate the magnification.
Okay.
I can give you the actual length. So the actual length is 45 millimeters.
Okay.
Good, 2 marks.
Okay.
Next question. Explain how the cells labeled A and the structures labeled B protect the lining of this trachea. So this is 4 marks.
So A.
So cells labeled A and structures labeled B.
So A are goblet cells. Goblet cells secrete mucus from their mucin droplets. Mucus is a sticky substance which traps pathogens and prevents them from entering the circulatory system. And the cells of the airway. The airways. B, cilia. The cilia beat up the mucus. Beat the mucus up the airway towards the larynx. In order to be swallowed. Yeah.
Okay, cool. You get the marks for that.
Okay.
If you want, I can show you how I would potentially do the answer for this. And then, so like you've gone through each stage, which is good. It's logical, and you'll pick up the marks for it.
Mm-hmm.
You could maybe save some time if you write it in more of a shorthand. So we know if you say A equals goblet cells and B equals cilia.
Do they accept that?
That
okay?
you could write like that, yeah.
Okay.
That you would get a mark for that, and then you can say, but then you have to explain. So you can say, you could have one bullet point say goblet cells produce mucus And then you have one bullet point that says mucus traps, which is, don't write it in that, so like that shorthand, but, that's perfectly fine. And then you can say, leave a line, say cilia, move mucus to the larynx. And then you could say that that stops pathogens from accumulating, et cetera, et cetera. What you did is fine, you'll get the marks. The only thing I'd be concerned of is either you might find it difficult to fit it all in the box, or you might push yourself for time when you're moving on. What you could always do is like sort of write a shorthandish answer like that, and then always come back, because I think you're bound to have extra time. Have you done any like sort of?
I normally like always have extra time.
Yeah.
Like at least 15 minutes.
Yeah. Okay. Cool. And have you practiced actually writing in these like
I haven't
allocated
written
spaces?
it in. Oh yeah, I have.
So you like print out the paper?
Yeah.
And okay. Cool. That's definitely important as well. Anyway, let's move on. That's good. Okay. Cells such as C at the base of the epithelium divide by mitosis. Describe the changes that occur within the cell between the beginning of prophase and the end of metaphase.
Okay. So prophase. Chromosomes condense. The nuclear envelope and the nucleolus are broken down.
Good.
The centrioles move to opposite poles.
Good.
Okay. And then the metaphase, the mitotic spindle made up of microtubules binds to the centromeres of the chromosomes. The chromosomes are aligned at the equator.
Good. That's perfect. You picked up every single mark that they've mentioned, and you kept it really concise. One thing that they talk about that you didn't say is referring to the random arrangement of the chromosomes at the equator.
Okay.
Literally, don't worry about it. That's fine. Do you know of random arrangement? So it's basically one of the ways that cells can generate diversity in their progeny. So in the daughter cells, one way that they can make them more diverse than the mother cell is by randomly arranging the chromosomes. And that means that when they're split, there can be, a sort of a slightly different complement in the daughter cell compared to the mother cell. And that's just because of slight changes that you encounter in A-level biology. I don't think it's really important at the moment, but basically when the chromosomes are recombining and thickening, mutations occur on the different chromatids. So with the random arrangement, you get a sort of addition of more diversity. But anyways, let's move on. That's really good. Any questions there?
No.
Okay, 2A.
Carbon dioxide is transported in blood in various forms. Describe how carbon dioxide molecules reach blood cells from respiring tissue, respiring cells. Okay.
Just gonna try and zoom in a bit.
So carbon dioxide diffuses down, from high partial pressure to low from respiring cells to the red blood cells. Carbon dioxide diffuses out of the respiring cell, crossing the cell surface membrane. The endothelial cells, the capillaries, and across the membrane of the red blood cell into the plasma, into the cytoplasm.
Good. You picked up all the marks again. Just saying, so this is 2 marks, and you have the potential of picking up something like 4. So you could pick up the 2 marks in 4 different ways. 1 mark for saying diffusion, so that's like underline it when you say diffusion. Whenever you say a sort of named process, it's useful to just underline it so the examiner instantly sees that and picks it up. So that's 1 mark. Another mark for saying down the concentration gradient or partial pressure gradient. Third and fourth mark can be picked up from describing the pathway which you did. So respiratory cell produces carbon dioxide, diffuses across its plasma membrane into the tissue fluid, then across the basement membrane, then across the endothelial cell, and then into the plasma, into the red blood cell. So that's fine. You get all the marks there. Any questions then?
No.
Good. Okay. Take a bit.
Well, blood has a low blood pressure. And why? It has a lower glucose concentration.
Good.
It has less white blood cells
Okay, why would it have less white blood cells?
because white blood cells enter into the tissue fluid.
Okay, I mean, it's a good thought. I think, like, unless you know that there's been injury, I wouldn't say, I wouldn't mention white blood cells, but you've missed one that you'll get maybe if you think about the way they've asked the question. So they say, like, other than the concentration of carbon dioxide, so
Oxygen,
yeah, okay, so what? More or less oxygen or what?
less.
Okay, perfect. So you get the 3 marks. There are a few other things that you can talk about. So you could say amino acids, you could say Y is more concentrated.
Okay.
Basically, if you talk about it and you give a little background, you'll get the marks. Why might there be more? So in what situation would there be more glucose in Y than X, as opposed to less?
Well, in this situation, because it's more concentrated, it has more glucose. So 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 it has more So why would there be more?
Yeah. So you could say less, you get a mark. You could also say there'd be more, but if you said there's more, you'd have to specify something. What kind of tissue would we have to be looking at for there to be glucose added to the blood?
Oh, it would be like in the digestive system. So
Could be, yeah. It could also be in the liver. So if the liver is receiving signals to release glucose, break down glycogen to produce glucose, then that could be happening there. So good. It's a bit more of a tricky question. Okay,
carbonic anhydrase.
good. Next one.
Okay. So hydrogen carbonate ions are the way that most CO2 is transported.
Okay.
It's transported back to the lungs.
Okay.
So the hydrocarbonate ions diffuse out of the red blood cells into the blood plasma. I think 85%
Good.
of carbon dioxide is transported this way.
Why do we have to do this reaction? Why don't we just transport carbon dioxide as it is in the blood?
Because otherwise it wouldn't result in the Bohr effect. So
Yeah,
it would
good.
result in more oxygen being released.
Okay, so another point there is maintaining the gradient.
Yeah.
And one final point, just another physiological point as to why you don't want carbon dioxide in your blood is because it decreases the pH.
Yeah.
So by, transporting it as bicarbonate, you reduce that effect because the hydrogen ions can be sequestered by hemoglobin. But yeah, good. You get 3 marks for that, so that's fine. You can have a look here at the parking scheme. So they say you'd get a mark for saying that the reaction occurs very quickly. I don't know how that's very relevant to the question, but anyways. Another one so this was your first point. Carbon dioxide is transported as hydrogen carbonate ions / bicarbonate ions. That they diffuse out of the red blood cells into the plasma. Yeah. So 1 mark there, 1 mark there, 1 mark there. I've already picked up all 3 marks. And then so that blood can transport more than could be transported as carbon dioxide in solution, / saying 85% of carbon dioxide is transported this way. Fine, that's a 4th one. And then the last 2 things is what we were talking about. Yeah. So. Okay. Take a look at this.
Okay. Okay. Right. The pulse pressure. Okay. Yeah. Okay. So for CO2, 1 kilopascal of partial pressure, it's equal to like 55,
Yeah, I'd say 54.
54.
But you get it for 55 as well.
Yeah.
And how about for 1.5?
For 1.5 be equal to 36. No, wait. 30, 32.
Okay, good. And what are the units?
Well, 32%.
Good. So those marks. So any question where you have to pluck a unit from the graph, use a ruler, and they usually accept like one box difference of error, but just to be safe, use a ruler. You've got plenty of time anyways. Okay, next question.
Okay. So carbon dioxide reduces the affinity of hemoglobin for oxygen. So by the hydrogen ions binding to the hemoglobin, it replaces the oxygen, and oxygen is released
Okay, good.
more than it normally would.
Okay, good.
Yeah.
And commenting on hemoglobin itself, what changes are happening to hemoglobin to actually when it's binding oxygen versus when it's binding hydrogen? Tell me a little bit about.
Do you mean like oxyhemoglobin to hemoglobinic acid?
Yeah. So why does it bind acid to a greater extent than it binds oxygen?
Because it has a high affinity for
Yeah, good.
hydrogen ions.
Good, you're right. So those are the kind of comments they want you to be making. So always, I know that you know this material fine. So when you're answering these questions, always think like, what sort of keywords can I fit in here? Things like affinity, things like conformational change. They're just like tick boxes for the examiners. But yeah, that's good. You get the marks there.
Full effect.
Good. Question 4.
Well, okay, so it causes more oxygen to be released. Thus, in respiring tissues, more respiration can occur because more oxygen is released.
Okay, so structure this into an answer for me. You got
Okay.
3 marks here.
So when carbon dioxide diffuses into the red blood cell, it produces hydrogen ions, which replace the oxygen bound to the hemoglobin, thus resulting in more oxygen being released.
Okay, good. That's like your basics. You've pretty much gone through this
I've already
before,
said that, yeah.
but you're still picking the mark, so it's important to go through it again. And then go on to say what you were saying. So why is that important?
The oxygen release is important because for the respiration of the respiring tissue cells.
And those cells are, how are those cells sort of driving this process?
Well, it diffuses from a high partial pressure to a low partial pressure.
What does?
The oxygen.
Yeah, but why is it that cells with a high respiration rate are sort of mediating the Bohr effect? What's the key here? So we're thinking of a bit of a cycle, right? Oxygen's getting displaced and going into the cells, and what's driving that?
It's respiration.
Yeah, and what's produced by respiration that drives that?
Well, respiration requires oxygen and produces carbon dioxide.
Exactly. Okay, so just like tie it together there with the fact that, okay, you've told me that carbon dioxide stimulates all this displacement of oxygen and it's used by the cells. Make sure you then say those cells are producing loads of carbon dioxide, so they're sort of feeding themselves oxygen.
Okay.
A form of auto-regulation. Yeah, good. So for this.
Okay. Um, okay. D is uracil.
Good.
E is adenine. Or adenine.
Mhm.
And then F is ribose.
Good.
Sugar. And G is phosphate group.
Good. That works. Okay, so maybe draw this one out and just fill in the table so I can mark it for you.
Okay. Oh, do you mean there?
Yeah, I mean, just do it on the screen, that's fine.
Um,
Good.
or I could do uracil instead of thiamine.
Yeah.
It's better.
Yeah, good. I would go with that as well.
Yeah.
Only thing, when you're talking about the helical nature of DNA, that's correct, but mention that it's double helix.
Yeah.
Okay, next one.
Okay.
So when you're starting this one, again, keywords that they're looking for. What process are they asking you to describe?
Translation.
There, you've already picked up the first mark, like literally just writing translation and putting a line under. I know it's really simple, but okay, tell me about it.
Okay, so translation. And so the mRNA binds to the ribosome. And okay, so T tRNA molecules bind to the ribosome and complementary anticodons bind with hydrogen bonds to codons of our mRNA. Two mRNA codons are subjected to the tRNA at a time. The ribosome moves along the mRNA, and peptide bonds form between adjacent amino acid molecules through a condensation reaction.
Okay.
And this determines the amino acid sequence, which is the primary structure, which affects the tertiary and secondary structure, thus the shape and function of the protein.
Okay, good, perfect. You get all the marks there. One hint, I guess, is if you're ever asked to do a big description like this translation, whenever you're talking about, codons, anticodons, tRNA, all these structures that you pick up marks just describing, it's sometimes useful to just be like, okay, mRNA goes to the ribosome. tRNA, which has an anticodon and an amino acid, also goes. The anticodon binds the codon, blah, blah, blah. It's just like semantics. You're still getting the marks, but it might help you structure an answer a bit faster that has a bit higher yield on the marks. Anyways, okay, let's do this one.
Okay. Hydrolysis.
Good.
Temperature of water. Okay. The temperature of water is raised to a boiling point in order to denature the enzyme sucrase, so no further sucrose is being hydrolyzed.
Good. So one mark that would say we want to stop sucrose being hydrolyzed, and another point saying that it's through denaturing the enzyme. And explain the results.
Okay. So,
Exhausting question.
yeah. Yeah. So, okay. So as sucrose concentration increases from 0 to 80, the units, the rate of enzyme activity increases. And then the enzyme activity stops increasing at 80 grams per whatever.
Okay, what's a word used to describe that?
Yeah, so Vmax is reached. Is that what
I mean, just describing the shape.
Well, it
of the
flattens, yeah.
Yeah, flattens or plateaus.
Yeah.
If you use those terms, just but anyways, cool. You got 2 marks there for describing it. Now explain it.
So, so for the first point, that it increases, the substrate concentration, the substrate is the limiting factor.
Good.
So, as the concentration increases, the rate at which the collisions occur increases, and more enzyme substrate complexes are formed at a faster rate, at a higher rate, thus producing more products or forming more products.
Good.
Then, so when the curve flattens, Vmax is reached. So this is where all of the active sites are full already, and the enzyme are the limiting factor.
Perfect. You've got every mark there. Pretty much just as they describe it here. So keywords there, limit is talking about the substrate being limiting factor at the start, and then the enzyme being the limiting factor. Quote your numbers as you did at the start, describe the plateau, etc., etc. You got it all. All right, shall we take a break there and then finish off this paper in 5 minutes?
Yep.
Okay, so different biological molecules.
Okay.
Complete this table by indicating which molecule matches each statement. Go ahead and fill out the table.
Okay. Well, I'll just
Yeah, just fill it out. Okay.
this one. Okay.
Good. All correct. The only ones where you had options are the one where it says used for an energy storage in plants. So you can talk about the protein or the starch or the M. And forms a helical structure, you could have also said the protein because that proteins can form helical structures. So
Makes sense.
secondary structures could be alpha. But good, it's fine. You pick up all those marks. Okay, infection of the day.
So, active immunity. Active immunity is where the antibodies are produced from oneself, and passive immunity is where the antibodies are foreign and enter your system. Active immunity results in long-term immunity, while passive in short-term. Active immunity to gain active immunity, an immune response is triggered, and passive immunity, there's no immune response triggered. In active immunity, you produce memory cells, and that doesn't happen in passive immunity.
Okay, so tell me a little more about the mechanism of active immunity.
So, antigen is recognized as foreign molecule, and then a lymphocyte binds to it with a specific attachment region and triggers an immune response.
Okay, what's that immune response? What does it actually do?
Primary response.
So when we got your antigen, you've got your lymphocyte. lymphocyte has a receptor that binds the antigen, what happens after it's bound?
It's activated. The lymphocyte is activated.
Okay, what do you mean by saying it's activated?
Well, it undergoes clonal expansion.
Good.
And it produces cytokines, which activate B lymphocytes.
Good, which also undergo clonal selection, and you've already told me about memory cells. That's fine, but you've got all that information put in the answer because they're literally just saying what's the difference between active immunity and passive immunity. Passive immunity, there's not much to say about it. Be careful about saying that it doesn't cause an immune response because technically, if I gave you antibodies, it would bind the antigen, but then the antibody would activate your lymphocytes, your phagocytes to get rid of the antigen. So it's technically still an immune response, but otherwise you're fine. Okay.
So what are the different points that I could have gone there?
Okay, so here you would you would have gotten a mark for saying that the immune system responds to an antigen in active immunity. Versus passive immunity, you're just giving antibody, which will respond to the antigen. Another mark for saying clonal selection occurs, / referring to B cells or T cells being activated. Another mark for saying that antibodies are made as opposed to just being given in passive. And then, this stuff that you picked up at the start of your response, memory cells are produced, long-lived versus short-lived. And then you could have picked up another one for saying non-immediate versus immediate.
Okay.
But if you were talking about it in the sense, so the way they phrase this question, they expect students to talk about active immunity compared to passive immunity. But if you compared it the other way around, you would have still gotten all the marks. And sometimes when students do that, they'll state things about passive immunity, such as antibodies are removed from the circulation, which is why it's short-lived, etc., etc. But that's fine, you get the marks. Okay, a bit of a trickier question.
Explain why the vaccine. Okay. So because the child receives or already has antibodies from the mother gained by natural passive immunity. So these antibodies could be IgA antibodies. And so antibodies are already present in the circulatory system.
Okay.
So there's no need for the vaccine.
Okay, what does the vaccine do? The vaccine it's not just injecting antibodies.
And well, maybe so the immune system isn't developed enough.
Yeah, good.
To form an immune response.
Yeah, good. So that's the key thing there. Okay. The vaccine is quite different from just giving antibodies, as you know.
Yeah.
So, you could say that the baby is not immunocompetent. It won't be able to bring up its own response in the first few months of life. But that's fine. If you read the question again, the last sentence kind of gives it away. Babies are born with a passive immunity to measles. So you're kind of told then that there's a placental transfer of antibody, right?
Mm-hmm.
So, I mean, you wouldn't get a mark for just quoting the question, but you get the marks anyways. Okay, let's work through this.
Okay. Okay. So
2 marks.
the 4 districts, I think, that have okay. 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 So the countries where, sorry.
It's okay, take your time.
Okay. So there's no pattern in the graph, or it's fairly random arrangement. But the countries with 90 or more percentage of districts have a very low mortality rate.
Okay, good.
Yeah, that's all I've got.
Okay, so if you say under 90%, there's a wide variation in death rates.
Yeah.
Whilst that's 1 mark, whilst over 90%, the death rates are very low. And if you go on to quote it, so like under 50 deaths per 1,000 children,
Yeah.
then you can pick up another couple marks. What mechanism are we talking about here, really? Why, if you were like WHO officer and you told me immunize 90% of the children, what mechanism are you trying to create? And good. Anytime there's like the whiff of a mechanism, write it down in your answer because you'll pick up a mark.
Okay.
So even just writing herd immunity will decrease transmission, pin that on, even though they're asking you to like describe the graph. You can describe the graph and then add that to your explanation why the WHO recommends it. Okay, that's fine. Okay, that's the end of that paper. Let's do the next one. Have you got it up? Yeah. Okay. You happy to go ahead to the next one? Do you want to do something else?
I'm fine.
All right, cool. So we're looking at the 2013 paper now. Okay, look at this. So before we look at the question, I want you to interpret this electron micrograph for me. So tell me what you see.
Okay. I see the 2 red blood cells.
Good.
I can see the endothelium made out of cells.
Good. How many
Single
cells?
cell,
Yeah.
thick.
Cool.
Very thin endothelium.
Yeah.
I can see. Well, I can't see, but I like has to be tissue fluid. around it.
Yeah, okay. And what do you see underneath the endothelium?
What do you mean?
So
Like the lumen?
the basal side. Okay, no, not the luminal side, but the basal side. So do you know there's a membrane that forms around endothelial cells called the basement membrane? And if you
So is that this?
that black line there, you see, it goes around sort of irrespective of the cells it exists, and you can usually always see it with electron microscopy. But yeah, those are the main components: endothelial cell, basement membrane, lumen, red blood cells. You might be given this without any context, and they ask you to identify what it is. If you can see that red blood cell, then you know it's a capillary. Otherwise, the structure would be way too small to be a vein or an artery. But anyway, good. And then tell me what they're pointing at then in A and B.
Well, endothelium. Well, endothelial cells.
For A, and how about for B?
It's a nucleus.
Good, yeah, that's correct. Are you happy, like, interpreting other like this? Yeah. Okay. All right, we just did that. We can skip this magnification thing. Explain how capillaries are adapted to the function as exchange vessels.
Capillaries have thin walls, single cell thin walls. This allows for small distance for CO2, O2 to diffuse across, thus resulting in a higher rate of diffusion. Capillaries have gaps, which allow the plasma to leave the capillary and form the tissue fluid. Yeah.
Okay, what does the size, what is the diameter of a capillary due to the blood flow?
So yeah, the capillaries have a small diameter and allows the red blood cells to be close to the respiring cells, thus decreasing the distance of which CO2 and O2 diffuse by.
Yeah, okay, cool. But in terms of flow, as in the rate of blood moving, what relationship does diameter of a tube have with the flow going through it? Any idea?
Well, if it's a smaller diameter, then there'll be a higher pressure. Okay.
Yeah, but there's a physical limitation on how much can be moving through it, so it's slower flow. So whenever you think about capillaries, it's going to be slower flow than, for instance, an artery or even a vein, because you can move more material. You just imagine the cross section.
So volume.
Yeah. So
that's why, like, when arterioles, when the smooth muscle contracts, then less blood is reaching.
Yeah,
it's
exactly.
flowing into the capillaries.
Exactly, you decrease flow. Okay. But yeah, there's loads of different points you can make there. So you definitely made 2. Okay, composition of blood, tissue, fluid, and lymph. Explain the differences for white blood cells, glucose, and protein.
Okay. So white blood cells. So the lymph has a high concentration of white blood cells because it passes through the lymph nodes. where a lot of white blood cells enter the lymphatic vessels, and the blood has a relatively high concentration of white blood cells, some of which can enter the tissue fluid to the gaps in the capillaries.
Okay. Going back to why there's such a high concentration of white blood cells in the lymph, tell me a bit more about that. Not relevant to the question, but I just want to test your knowledge.
Okay. So the lymph passes a lot of the fluid lost in the capillaries, and it returns back to the veins.
Yeah, good.
But it also contains pathogens and other invasive microorganisms. So it passes through the lymph nodes where there's a high concentration of lymphocytes and phagocytes. And antibodies are secreted into antibodies leave the lymph nodes, enter the lymph.
Okay, so it's kind of like a completely separate circulation. As you know, most of the fluid that comes out capillaries and goes into cells will go back into the
But like 10%.
flow. But yeah, you have this small proportion that's not going into the blood, and a lot of that is cellular debris, and not coincidentally, a lot of pathogens and pathogen proteins, which sort of come along with cellular debris, are going to be blocked from getting back to capillaries.
Mhm.
And so the only place they can go is into the lymph, where they're trafficked through this system of nodes. And as you say, the lymph has a very high concentration of white blood cells because it's in the lymph nodes that clonal selection happens. So white blood cells will pick up these bits of debris. So now we're talking about lymphocytes. They'll process them and try and put them on the receptors if they bind, and then they can be clonally selected in the lymph nodes. And then lymph nodes are also connected to the blood flow. So via lymph nodes, clonally expanded B and T cells can be secreted to the blood flow.
Yeah.
Okay, we've gone over white blood cells. How about glucose?
Glucose is a relatively small molecule, so it can leave the capillary by entering the gaps in the capillary.
Mm-hmm.
It can diffuse.
Yeah, okay, good. Keep
and it diffuses into the lymph from the tissue fluid.
Yeah, okay, good. And protein?
Protein are larger molecules. So plasma proteins are too big to enter through the gaps, to diffuse through the gaps into the tissue fluid, which is why there's so much of a higher concentration of the protein in the blood compared to the tissue fluid.
Okay.
And protein is returned by the lymph into the circulatory system.
Yeah, what kind of protein might you find in the lymph? What kinds of protein?
Well, you could find antibodies.
Yeah, good.
Protein.
Good. Or broken down pathogens, cellular debris, that kind of thing. But yeah, good. Okay, we'll skip this one because we've pretty much gone over it in the last paper. Okay.
Measles is transmitted by droplet infection. So an infected person sneezes / coughs, and an uninfected person inhales the infected droplets, thus the disease is transmitted.
Good, okay.
By the mobility virus. No, wait, yeah. Okay.
Good. Now moving on. How are we doing for time? Okay.
So the variable regions form an antigen receptor or antigen attachment site.
Okay.
And so each antibody has a specific antigen receptor with a specific shape, complementary to certain antigens.
Okay.
That's not yeah. B are the disulfide bridges, which are part of the quaternary structure of the protein and will stabilize as the protein holds the structure together.
Good.
C, the constant region of the heavy chains. So they bind to phagocytic receptors, which have a complementary shape to the antibody region and allow for phagocytosis to occur.
Good. Good. You get all the marks there. Just one thing I would say, actually, when you're talking about the variable region, be careful when you use the word receptor because, in fact, I would avoid, if you're talking about the structure of an antibody, just don't use the word receptor because none of this is a receptor. You have antigen binding site here and the constant region, which tends to be a receptor binding site. And they can sometimes get a little bit finicky with that. Okay, transpiration.
Okay, so stomata open in order for CO2 to diffuse into the plants. And CO2 is the source for carbon for the assimilates formed in photosynthesis.
Mm-hmm.
And the stomata being open for CO2 results in transpiration occurring, so results in water vapor being lost by diffusing down the concentration gradient out of the plant.
Okay, perfect, good. All right, this might be one of those. Let's just, oh, this is fine. 5 marks here to describe the results of this experiment, so I'll read.
Okay. So both transpiration rates of the sheltered and exposed location increase. Then they peak twice, they decrease again. So both of them peak at 12 and 16. 12 and 16 o'clock. And the values for the transpiration rate, like I will code them too. But yeah. And so the increase of the transpiration rate of the sheltered location has a steeper gradient than the one of the exposed location. The same for the decrease. And so between the two peaks at 14 o'clock, the transpiration rate for both of them decreases. The transpiration rate of the sheltered location is higher than that of the exposed location, from 0 to 24.
So it's always high.
Yeah, it's always high.
Good.
And what else? Yeah.
Okay, so I like your structure. You started by just telling me like the similarities, and I would do that. Always start with similarities and do the differences. The more structured you are with this, the less, unnecessary words you can put in your answer. The only thing you missed to comment on is the fact that both of them shut down at night. So between around about 8 p.m. until around about 4 a.m. they're both pretty shut down, which is relevant to transpiration, and it's probably going to come up in one of the next questions. So mentioning it is always worth it, but you will pick up the marks there. These are the really, as you know, laborious questions where you just have to describe what you see. Okay.
So one would be thick, waxy cuticle. It increases the distance over which transpiration occurs.
Okay.
Another one are hairs. So hairs outside the stomata reduce the water potential gradient by trapping water vapor. Yeah. Another one would be sunken stomata, which prevent wind from maintaining the water potential gradient.
Okay, perfect, good. They're all valid, they're all right.
Do I have to did I only have to state 3 things?
They-
not explain it? Or
They are basing their marking scheme on descriptions, although having said that, some of them are just statements. So identification of the following features: stomata closed for longer during the day when hot / when dry. Stomata in pits / sunken stomata, but the question is saying describe, so
Describe.
don't risk it. Good. Okay, we've got 2 base pairs of the DNA molecule.
Yeah.
In the bases J and K and the bond labeled L.
J thymine, K guanine, L hydrogen bond.
Okay, good. Okay.
Okay, so words that we're looking for is complementary and non. Sorry. Competing and non-competitive and non-competitive.
Good.
Enzymes.
Really important there. Yeah. First two bullet points.
So cytopidine is competitive enzyme, while efavirenz is a non-competitive enzyme. So the cytopidine, competitive enzyme has a similar shape to the substrate. Thus, it binds to the active site and inhibits enzyme action by preventing substrates from forming enzyme-substrate complexes.
Good.
Non-competitive inhibitors bind to allosteric sites, so
Good. Yeah,
sites
that's the
other
one. You need to.
allosteric.
Yeah, go for it.
And they cause a change in shape of the enzyme. Thus, the active site, the shape of the active site changes as well, which results in the substrate not fitting into the active site, thus inhibiting enzyme action.
Good. Perfect. The only two marks that they've referenced here that you haven't gone over is like, it's sort of, I don't think it's fair to expect them to, like, for them to expect you to come up with this because they're not really asking it, but they say if you had zidovudine, you would be able to reverse its effects by increasing the concentration of the substrate because it's a competitive inhibitor, whilst with efavirenz, that wouldn't happen because it's a non-competitive inhibitor. But that's fine. You pick up the marks anyways. I think it's just for people who just put the information in. Anyways, people who receive drug treatment for HIV, take a mix of drugs, just the advantage.
Okay. So it's less probable that the enzyme, that the virus is resistant to all drugs. So that's the used in combination therapy.
Okay, so virus might be.
Yeah, so the virus might be resistant towards one, but it's less probable that it's resistant to all of the drugs.
Yeah, okay, and explain resistance here to me.
So resistance is because by mutation of the DNA or genetic information of the virus, causing a change for protein or whatsoever, thus gaining resistance.
Okay, so what protein is being changed here for these drugs specifically?
The reverse transcriptase.
Yeah, good.
Or protease.
Yeah, cool. And what parts of the proteins are being changed by the mutations that make the HIV resistant?
The active sites, or well, it could be no, it can't be actives. Well, yeah, it's an active site because otherwise the competitive inhibitors can't bind to it.
Good. So it's either going to be the active site for the
Or it's going
competitive,
to be an allosteric site
for the non-competitors. Okay, good. Now you've sort of explained to me the mechanism. Now, why is it advantageous? So, from a more like public health perspective, why are we doing this?
Well, do you mean like if we weren't to treat people with multiple drugs, the virus would, there would be more of a chance that it would develop resistance? Because it either stays in the body for longer or there's just more of them.
Yeah, and so you might not know this, you probably do. HIV has a high mutation rate. Okay, so public health perspective, you can either spend a lot more money on giving those people combination drugs, or you can try and deal with the consequence of HIV strains in your population being highly resistant to some of the drugs. So the answer you could go for here with that respect, don't worry about it, you've picked up all the marks anyways, but I'm just trying to like quiz you on these perspectives. The mark you can pick up is saying by trying to hit all humans with the infection with all these drugs, you can prevent resistant strains from occurring and therefore being in the population. And if you have resistant strains in the population, it is much harder to, and it can be impossible to deal with them. I mean, literally, if you had one individual who had HIV and you sort of cultured a resistant strain of the virus in them, you could end up causing like huge problems for public health in wherever you are. So yeah, it's important from that respect too. It's kind of linked to the fact that you're trying to prevent the virus from becoming resistant, but resistance is hugely important to public health as well. Okay, good. Last part of this question then, antibiotics. Take a break.
Antibiotics are not effective against viruses. So, okay. The HIV has different proteins, and it uses the metabolism of the host also to replicate.
What do antibiotics target?
So antibiotics target like the cell wall or DNA replication. Yeah, different types.
Yeah,
The viruses don't have cell wall.
Yeah, that's a mark there. Viruses don't have a cell wall. But yeah, it's I don't think it's like the best worded question, but essentially you'll pick up marks for explaining how antibiotics work and how they wouldn't therefore work on
So
the
they
viruses.
like kill or slow growth of bacteria without harming the cells of the human cells. So
Good. Okay, so not part of the question, but another sort of public health thing that you could tell me about. So theoretically, why wouldn't we just give antibiotics anyways to everybody with HIV and AIDS, just so that when they, they're immunosuppressed, it's likely they're going to get of bacterial or fungal infection anyways, because they're immunosuppressed. So why don't we just give them all antibiotics all the time anyways, even though we know it's not treating the HIV and the AIDS, but it could, for instance, reduce the pneumonia and the urinary tract infections and that kind of thing.
Because antibiotics should only be prescribed when necessary.
Why?
Yeah. Because there's a higher chance of resistance developing against the antibiotics. Yeah.
Okay, so you've got an individual with HIV, you've given them far too many antibiotics, and they start growing resistant bacteria. What's the problem? What's the public health problem there?
Well, all of the antibiotics that they've given out to people don't work anymore.
Exactly, yeah. So this person who is cultured a resistant strain can very easily pass it on to other people and who might not be immunosuppressed, but for whatever reason the infection takes hold, and then it could be something that's easily treated with over-the-counter antibiotics, but not anymore because it's resistant. The person has to end up going to hospital. Potentially there's no treatment available, so we're aware now of some animal bacterial infections that there have been reported cases where they've transferred to humans, in our widest spectrum antibiotics that hit everything we know how to hit doesn't touch the bacteria, and the individuals end up dying. So it's already started, this like pan resistance has already started to emerge. In terms of resistance, like other than patients getting prescribed too much antibiotics, what else drives antibiotic resistance in the world?
Okay. So when they don't finish their treatment, so they think that the symptoms are gone, so they just finish antibiotic treatment, but not all of the antibiotics, all of the bacteria is like killed, so it lingers on in the body. Can also be from so people in certain countries, antibiotics don't have to be prescribed. So that's also something that could cause. And a lot of people have antibiotics. So instead of going to the doctor, they just take those antibiotics. That's awesome. And well, we already kind of said this, but antibiotics only when necessary. Take that. And
you've given me all the points that I can reasonably expect about human interactions with antibiotics. How can we create antibiotic resistance in environment?
So by using it in farms.
Yeah, that's such an important reason.
Yeah. Yeah.
So tell me a bit about that. Why is it used?
So they, together with the food that we give animals, the antibiotics are used. But that can grow a culture of resistance. Like hospitals sometimes, if there's too much antibiotic use, then could also.
And what's the incentive for farmers to use antibiotics? when they're feeding their chickens or their cows?
I think like it helps them like the metabolism or something like that. So
Yeah, so
they grow at a faster rate, stuff like that. So it's not necessary, but they still do it.
Good. So it has nothing to do with protecting chickens against bacteria. It's everything to do with yield. So they're very focused. If they spend £1 million on 10,000 tons of antibacterials, and that means they increase their gains from chicken meat by 10 million pounds, and they'll do it. There's no problem. The only thing that stops them then is regulation. So in the EU, you're not allowed to give animals antibiotics unless it's to treat infections.
Yeah.
But those laws don't exist in huge countries which manufacture a huge amount of meat, like China and other areas in Southeast Asia, and also the States. So yeah, farming, huge problem. One last thing then on antibiotics. How can a bacteria develop a resistance mechanism to antibiotics well before like humans ever started using antibiotics, well before humans even existed? So some of the resistance mechanisms, if you look at it on an evolutionary tree, they're millions of years old.
Because so I know about so it can occur through horizontal transmission.
Good.
From one species to another species. So there were pathogens which were constantly like in the soil, right? And surrounded by like different foreign molecules. So if they gain resistance, they can transmit it to like another species of pathogen by horizontal transmission.
Okay, cool. Yeah.
But also through vertical transmission.
Good.
Which is just generational.
Okay, that's good. But in terms of how the first instance of resistance comes about, how can that first instance in that one bacterium happen before humans existed to be putting antibiotics on it for the bacteria to have a selective advantage to
Well, do you mean like through random mutation?
Could be. That's one possible. Could be random mutation. Where do we get the antibiotics from?
From fungi and bacteria.
Yeah. So what?
That's what. Yeah. So fungi have been like a lot of the resistance comes from fungi.
Yeah. So you can imagine a sort of arms race, biological arms race between bacteria and fungi that predates human, predates mammals, predates most living things. So they've been developing intricate resistance mechanisms, and just because we've been able to isolate the antibiotic from the fungus doesn't mean that the bacteria hasn't already developed its own way to resist it.
Yeah.
So that's one thing. Ways that we can get around that is by adding synthetic chemicals to that antibiotics.
Yeah, and causing. Yeah.
Or you can take this sort of chemical approach to producing antibiotics that completely, you put all your fungal antibiotics aside and you just start with a test tube and a bunch of reagents and you come up with an antibiotic. And those ones, and we have a few, I think there are 3 or 4 classes that are used. It's really difficult for bacteria to develop resistance mechanisms to those because generally resistance mechanisms can take a very long time to develop in bacteria. Viruses, not so much, because they have such a high mutation rate that random resistance mechanisms occur. With bacteria, it can be a bit more difficult. Okay, that's good. Sort of went through that. Shall we take a quick break then and then finish this paper?
Yeah.
Cool. Have you had Okay, I think it's just a couple more questions. So, cell surface membrane, functions of P, Q, and R.
Okay. So P is a transport protein, which can provide it's a carrier protein, and it can pump hydrophilic molecules and ions through the membrane against the concentration gradient by active transport.
Good. Yeah.
Okay. Q. That's the glycocalyx. It stabilizes the membrane by forming hydrogen bonds with water molecules. Together with proteins, it forms receptors.
Good.
Cholesterol maintains the fluidity of the cell surface membrane.
That's perfect. You don't need to explain W. That's perfect. Okay. Circle the width of the membrane shown as W.
7 nanometers.
Cool. Membrane surface, cell surface membrane is kind of fluidizeic. Okay, so very standard question.
Yeah. Okay, so fluid is due to the lipids and certain proteins being able to diffuse throughout the membrane, and mosaic comes from the irregular arrangement of different sized proteins within the membrane.
Perfect.
Okay. So aquaporins allow for facilitated diffusion of water molecules down the concentration gradient.
Okay.
And they allow for a higher rate of exchange of water molecules than water molecules diffusing through the membrane, because it forms hydrophilic regions through which water molecules can be exchanged. This could be important for regulation of the water potential of the cell and surrounding environment.
Okay, why is it important in plants and why is it important in animals?
So for animal cells, it's very important because if the water potential isn't maintained or regulated, this can lead to bursting of the animal cells or shrinking. Plant cells have cell walls. So
okay.
it's less simple.
Perfect. Explain how uncontrolled cell division can result in cancer.
Okay. Uncontrolled cell division by mitosis forms an irregular mass of cells, aka tumor. This tumor can interfere with the function of the tissues that it's surrounded by, and it can induce lymph vessels and blood vessels to deliver nutrients to it. which helps it grow. And it spreads through metastasis by part of it, part of the tumor entering the lymph or blood vessels and forming a secondary growth
Yeah, okay,
in the body.
good. And in terms of the genetics, what leads to this?
So, proto-oncogenes, or tumor suppressor genes, through mutation, which causes those genes to change into an oncogene. Yeah.
Good, so proto-oncogenes are switched on or tumor suppressor genes are switched off.
Sorry.
So, let's see. So you happy with the concept of proto-oncogene versus tumor suppressor gene? Let's say we have a cell cycle here. I won't draw all the stages, but you're a normal dividing cell. To go into mitosis and divide, you have various checkpoints. And you have genes which encode proteins which will drive the cell forward. And those genes are your proto-oncogenes.
Yeah.
And you have genes that at the checkpoint will say, we need to stop because something's wrong. And those are your tumor suppressor genes. So both of us have both proto-oncogenes and tumor suppressor genes, and they're active in all our cells at varying points just to make sure that everything in the cell cycle is going on okay. If you mutate that into an oncogene, it's described as an on mutation because you essentially cause that gene to be regulated all the time, which is constantly telling the cell to divide.
And the other one's not.
And the other one is an off mutation, which just completely knocks it out and stops the checkpoint from stopping the cell. Okay, so that's what I mean by on and off. That's fine. Your answer is fine. You pick up all the marks. Experimental evidence that smoking causes lung cancer. Okay, go for this one, then we'll stop.
So, epidemiological evidence that shows a correlation between smoking and lung cancer. So people in workplaces that smoke more results in increased lung cancer. People who are in contact in like more smokers, like if somebody's part of a family where their parents smoke a lot, if they tend to have if the percentage of those people of them having lung cancer is higher than other people.
Okay, good. In terms of animal experiments, do you know any sort of experiments that could show that smoking causes lung cancer?
Was it with like, it's like with a specific animal,
Yeah.
is it not? With the dog?
Yeah, good. So it's a beagle experiment. So just give me the key points from that.
I think they, no, I actually don't know.
Okay, so there's three groups. There's three groups in this experiment. Dogs that smoke unfiltered cigarettes, dogs that smoke filtered cigarettes, and dogs that don't smoke. Obviously, the ones that don't smoke don't develop cancer. The ones that do smoke develop cancer, and the ones that smoke with filters develop pre-cancerous changes. And those are three different groups, three different marks that you can pick up. But you get the marks in English, don't have units. Okay, that's good. Let's stop there.